So, just a few words, Ashley. I'm very happy to welcome Professor Ashley Behrens today from the Johns Hopkins Hospital in Baltimore. With his extensive experience in ophthalmology for more than 30 years, Professor Behrens is a specialist in the medical and surgical management of cataracts and corneal diseases, including corneal transplantation techniques for Fuchs dystrophy, keratoconus, as well as LASIK, PRK, and refractive exchanges. We know each other since 1996 from Erlangen. He originally was born in Venezuela. In 2003, Professor Behrens joined the Wilmer faculty in Johns Hopkins, served as an executive medical director at the affiliated King Khaled Eye Specialist Hospital in Saudi Arabia, and is now associate professor of ophthalmology at the Johns Hopkins University and chief of the comprehensive ophthalmology division at the Wilmer Eye Hospital in Baltimore. Now, I have this thanks to Frau Turner. She prepared that very nicely for me. So I would like to introduce your talk, Ashley, International Keynote Lecture, Photodynamic Therapy for Infectious Keratitis. It's your turn.
Thank you very much, Bertolt. I really appreciate all the hospitality. Of course, I feel like I'm at home, and in fact, I'm at home when I'm with Bertolt. But I have really, really good experiences and thoughts from the time that I had my fellowship in Erlangen. I used to speak some German. Now it's almost gone, but I was able to order some bread in the bakery, a couple of days ago. So I was really proud of myself. And I could pay and everything else. But I really thank you for the kind words and the kind invitation. And I'm going to start talking specifically of this work that we have been doing for many, many years, actually, probably 15 years. So far. And it's PDT is photodynamic therapy for infectious keratitis. It was a very nice talk, Dr. Roth, that you had before, and I'm going to talk about a little bit about this because this seems not to be a good option for candida keratitis. I'm going to show it later. But for the other fungal keratitis, it seems to be pretty efficient. I have to say that I have financial interest in this, all the presentation. We have been working on patents and protection for this, and we're hopefully going to get this into the market as soon as possible. So we're going to be talking about the first steps historically, the riboflavin and UVA cross-linking that we started working with in 2005. Actually, it was the early stages of cross-linking, and we came to this by chance. We wanted to do some therapy to disinfect another option that we were using for eye drops. There was a biological material, and we found out that there was a company in the United States and actually in Baltimore that was producing a technology using riboflavin and UV light for disinfection of blood byproducts. So at that time, we said, well, why don't we do the same thing with the cornea? And now it's been used as cross-linking, and we started doing the trials, and voila, we found that it did work also for, in the cultures in the lab, worked for bacterial keratitis. And we started giving lectures. Of course, we didn't have any permission from FDA or anything like that, but we gave some lectures of the early results. And one of these lectures was in San Jose, Costa Rica. And in 2007, I think this is the first time that this therapy was used in a patient. We gave a lecture there. We talked with the people back home in Hopkins because we had a patient with acanthamoeba that one of my colleagues had there, and we decided that it was a potential good option. So we tried to do all the paperwork very quickly between the United States and Costa Rica, and we treated this patient. They treated that patient with the cross-linking procedure, and it worked. Of course, we had some work done in the lab before, and the cross-linking was already deemed as safe in Europe, and we were shocked with that. So we continued doing more studies. We organized the first ARVO, a special interest group in 2008 in ARVO, And we had a very strong presence of Germany. We have Klaus, who's not with us today. Unfortunately, but Klaus Grussiefen was part of that course, and it was a full house because nobody thought that this thing, cross-linking could work for bacterial keratitis. So, the whole story followed, and the first 3 patients treated in the United States were treated in our group at Hopkins at Wilmer Institute through an FDA exempt, and they were successful. So that was the first therapy that was used in patients in the United States. Wait a minute. So what are the advantages of this technology? Well, of course, this might overcome the testing. So you can start with that because it's a broad spectrum. So if you don't know exactly what is going on, this potentially could kill bacteria, could kill fungus. Not so much viruses. Actually, it might be detrimental for viruses in the theoretical standpoint right now. So I would hold off on that for the moment. But for infectious keratitis from bacterial and fungal and parasitic might be a good option. The antibacterial efficacy for this therapy has been attributed greatly in part to molecular oxygen. And this was a little idea that was given to us by one of our co-workers, who's Dr. Casadeval, who recently was elected as a member of the Academy of the Sciences at Hopkins, which is a very big honor. And he suggested that, Ashley, why don't you use oxygen? And we said, well, let's try it. And we started studies in experimental studies. And the idea or the question was how to give oxygen during the cross-linking process. So we started the basics. We started with, just blowing some oxygen in the plates to see what happens if we could do it in a cornea, blowing the air while we were doing the cross-linking, and didn't work. So we did some other trick was to oxygenate the riboflavin component, and we put some oxygen in the solution to try to increase the oxygen content in the solution. We did the experiments as well, didn't work. But we thought more in basic chemistry, and we went back to our high school studies to try to develop some sort of like source of oxygen. And then we decided to include some hydrogen peroxide in the solution. And voila, that was amazing, the result that we got. Now the thing is, what is the concentration? Of course, I'm going to be very broad here, not giving you specific details because we're still in the process of protection. But we started checking for studies which have been using hydrogen peroxide for contact lenses solutions, and we started with that. The concentration that was deemed as safe in contact lens solution, because you use it in the cornea and use it on a daily basis, and potentially the hydrogen peroxide at that concentration would be good. So, and it did work very well. So we started working at the microbiology lab at Hopkins, not at Wilmer, but at Hopkins, so the whole hospital, and they had the most feared strains of anything that you can imagine. So they had this strain of Staph aureus that was super, super resistant. And we did the first studies. And as you can see here, the untreated, that was, of course, in plates in the lab. The untreated, you have full colonies here, so it didn't do anything. The riboflavin with UVA, we treated this strain, nothing happened. With gaseous oxygen, nothing as well. Well, a little bit of effect, putting the flow of the oxygen in the plate. The oxygenated didn't work well, but the B-Parox, we call it, it was amazing. So it basically killed all the colonies there. And then we started playing with all kinds of bacteria that they had there that were super resilient. Some of them were obtained from patients that were actually dead from infections at Hopkins that was so resistant to multi-drugs, and we played with all of this, and they were very, very potent for that. They were effective for those. So we then thought, well, let's do an in vivo study, and let's do it in rabbits. Of course, when we're working with rabbits, probably here as well, to harm the rabbits is a big deal. So we spent a lot of time doing all the paperwork to try to do these studies in rabbits, because of course, you're doing one eye and you're creating a keratitis, which is not a nice, view in terms of the harming the animal. But we got approval. So, and we studied a very, very resilient bacteria that is called persister bacteria. It is a lab bacteria that is normally a strain that is very resistant, but we put them into several culture media so that it reaches a stage that is stationary, so it doesn't duplicate anymore, and that is virtually impossible to kill even with antibiotics, because the bacteria is, the superbug. So this is pretty much the characteristics of these bacteria. They're highly resistant to antibiotics. They create biofilms in the normal status. There's a major cause of antibiotic treatment failure, and this is a model designed to overcome the rabbit's immune activity. So we were trying to make sure that the rabbit's immune activity was kind of out of the study here. And this is what we did with 36 New Zealand white rabbits. We used 4 different groups, one with Behrens and UVA, the other group with riboflavin and UVA, the other group with conventional therapy with moxifloxacin. Hourly, poor fellow, she had to go and do the drops every hour for 6 days. And the untreated, we didn't do anything with them. And this is a little video that when she was doing the setup with the animals, and this was the keratitis. And you see here, one second, here that you have after the treatment was started 6 days after the established keratitis. All the rabbits developed keratitis. What you do is you inject the bacteria directly into the stromal cornea stroma, and that develops a very wild keratitis after 6 days. We did kill the rabbits at 3 hours, 24 hours, and 12 days after the exposure, and we also did clinical assessment for the surviving animals, including photography. This is how it looks. So the Staph aureus multi-resistant bacteria. This is at 24 hours. So you see the injection in the center. And after 4 days, you see a very well-established keratitis there model. Now, these are the results after the Behrens and the UVA. So day 6 after the treatment, you see that the infection is confined to the area. It didn't expand. And at day 12, and this is real pictures. This is not Photoshop or anything. This is what happened. So basically, of course, we're talking about rabbits. It's not humans, but it was complete regeneration of the cornea. This is a couple more cases of the group of the treated with Behrens and UVA. And the corneas were crystal clear after 12 days of the treatment. These are the ones that we didn't do in the groups. Of course, this one, we selected the worst ones. I have to disclose that. But this is 12 days after riboflavin UVA, the standard cross-linking, terrible cornea. And this is the 12th day after moxifloxacin. The histopathology was amazing. I mean, this is not a normal cornea. This is a real cornea after the treatment. So it's practically intact after 12 days of the Behrens and UVA. The important thing is this is just one application. We didn't do serial applications, just one application. Apparently killed everything. And you see that the endothelial cells are still there. So there was no damage to the endothelial cells. Of course, the rabbits have more resilient, and they regenerate the endothelial cells. So that's probably not the best model to see toxicity in endothelial cells. And this is the 12th day after cross-linking on the right-hand side. So you see vessels, a lot of inflammation. We also measure, in terms of safety, the apoptotic cells in the corneal tissue after 12 days of the B-Parax and UVA. And you see that the apoptotic cells in the B-Parax are really, really low as opposed to the cross-linking that didn't show major changes compared to the controls, and the no treatment. And these is the apoptosis of the immunohistochemistry after 24 hours. So you see some, positive cells here that were cells that were going to naturally die after the procedure and after the infection. And these are after 12 days. So basically there was no cells, apoptotic cells. So the cornea was back to normal. And these are the quantitative data. So you see at the very beginning, of course, a lot of apoptosis because you have an active infection there in the B-Parax plus UVA. But after day 12, it goes almost zero. Of course, it can't be zero because that's not the natural way it works. But you see with the other treatments and the untreated rabbits, the apoptotic rate was really, really high. The microbiology, we also cultured the bacteria after we got the samples, and you see that after 12 days, 0 colonies in the samples of B-Parax with UVA. The cross-linking one, some colonies there, less, but some, and the untreated, of course, was the highest. So with this little in vivo trial, we demonstrated that the treatment was with B-Parax and UVA was the only group that really showed clinical improvement. We also observed that there was apparently no damage to the natural normal corneal cells in those groups. And we think that B-Parax plus UVA might help in the wound healing process because corneas were dramatically good, regenerated after the treatment. And the B-Parax with the bacteria, with the persister bacteria at 24 hours killed, this very, very resilient strains of bacteria, which correlated with the in vitro studies. So that was a good correlation between the two. Now, we wanted to make sure, again, that the treatment itself was safe in terms of preserving the natural cells in the cornea. And we did 2 studies with specific markers for cell vitality. One is the LDH, and we did it in macrophages. So we did a culture. We treated these macrophages with the same parameters that we were using with B-Parax and UVA. And the way that this works is that the LDH levels tend to go up when there is cell destruction. The LDH is an enzyme that is inside of the cell. When you measure it outside of the cells, it means that there's a big level of damage of the cells because they break the membrane, and then you measure a lot of it. We didn't find any statistical differences between different treatments, the B-Parax with UVA, the B-Parax alone, vitamin B2 alone, vitamin B2 with UVA, cross-linking together. So basically, there was not such a big difference, and the untreated, of course, here. So there was no major damage to the normal cells with this assay. And we did another one, which is the ATP. So we measured the amount of ATP in the cells. So when the cells are alive, you have constant and good numbers of ATP. When the ATP goes down, it means that the cells are dead. So we did the cell vitality studies, and again, there was no difference in any of the groups. So we think that that treatment is definitely safe. Now, we continued testing some other strains for efficacy, and you can see here we did Cryptococcus, which is very resilient to a standard antimicrobial treatment, and it worked beautifully. These are plates, and you see the cultures here with the treatment. It seems like it was also effective with some of the components of the solution, but of course the Behrens with UVA was the most effective one. So you see that with one dilution of the supernatant that we get from the treated sample, it's very low the amount of colonies that survive. That means that it's very efficient, the treatment. And of course, the untreated, you see that even diluting the samples, we get a lot of colonies there too. So that's with Cryptococcus, and this is with Candida. With Candida species, unfortunately, it wasn't that great, but we're working on it. So there's a little bit of tweak that we're doing, and we were able to get very good results. Now we're testing the safety in further studies in vivo. But it does work as well, but not with the standard concentration that we were using before, as you can see here. There was some effect with the Behrens, but not dramatic. So we did some tweaks, and it improved. This is with Candida albicans, so albicans, right? Unfortunately, we have like 1/3 of the corneal infections here in Deutschland with Candida species, so this might not be a good option for those. The interesting thing is that this is not. You have antibiotics, and the antibiotics usually work for something. This kind of antibiotic is working for many different things, but not for all. So you have to be careful with that. I think in the future, when this treatment is really established, we're going to have antibiograms as well to see which species are more resistant to the treatment and which ones are more sensitive to the treatment. So we're going to have that kind of data available. And this was a nice thing that we wanted to show. It's a fungi that is basically the super fungi. There's no treatment known for this. There's some antimycotic solutions that have been used, but these are samples recovered from dead patients. You get this, even if you're immunocompromised, it's a death sentence. So my fellow, when she was working with this, she was really scared of having the proper safety measures, because unfortunately there's no treatment. So there's a high lethality rate with this when you get infected. It was first described, I think, in 1990 in Belgium. This fungal Lomentospora prolificans. We get the strain from Brazil. And it is, the Behrens with UVA is highly effective. If you see here, it kills completely, well, almost completely, the cultures that we had. So Dr. Casadeval was working with this specific strain, and he was impressed that he had something, at least for this particular treatment, for particular fungi. So the new and improved PDT, this has been years, 15 years of work. And, Bertold, sometimes he gets really bored with me talking about the same thing, for 15 years. But we keep working, and we keep improving things. So the Behrens with UVA appears to be safe, number one. Has a broad range of action against multi-resistant bacteria. The additional benefits in wound healing are currently studied, and definitely we see that there might be some additional effects in that regard, especially because of the cross-linking perhaps, and also to limit the multilateral or the collateral damage in the tissue. The reduction in treatment costs are going to be amazing, because of course the faster recovery of patients is going to help with that. And unfortunately, we have limited efficiency with Candida species, as I said before. So a lot of thank yous to my team. Of course, they have been in different time frames working with us. And Arturo is probably our closest collaborator now, and Dr. Zhang in the microbiology lab. So thanks to all. And of course, thank you for the warm welcome back to Germany and to your hospitality with my wife and myself while I'm here. Thank you so much.
So Ashley, when can we use that for patients?
We have to ask FDA. So that means, probably in a few years. But hopefully we're working on that.
Micro? Professor Gerling?
Congratulations, Ashley.
Thank you, Bert.
It seems as if you're down to something that might be helpful, not only for us, but also on a worldwide level where patients will benefit in the developing world.
Thank you.
I have two questions. One is a basic one and one is clinical. The basic one is, it would be even better if this would work without a cross-linking device. And I've not seen any control group in your experiments without cross-linking. So is the cross-linking, the UV light, mandatory or obligatory to achieve an effect?
Yes, that's an excellent question. At the very early stages, we tried it. So we tried to do the activation before and use it, kind of in light-protected bottles and tried to give the treatment like that. We tried also with natural light, natural UV exposure, and it wasn't as effective. So apparently there is a need of the light for the activation. Apparently what happens is that riboflavin decomposes into different fractions. One is the lumichrome, and the lumichrome acts as a nucleotide, and it goes into the DNA and disrupts the DNA. So that's kind of the basis of the treatment with the riboflavin. But on the other hand, with the oxygen, what we generate is a lot of free radicals, oxygen radicals, and that's really toxic for bacteria and for some of the parasites. Unfortunately, the free radicals and that's why I was telling you that I'm probably not very excited with viral. It might actually be beneficial for viral replication. So we are a little bit more, slowing the rate in terms of recommending this for viral infections. But unfortunately, it needs the UV light.
And second clinical question is, I suppose this depends on case selection as well. If you have a very severe keratitis, the light will not reach deeper layers and will be sufficient.
Correct.
Do you have suggestions? Is it only good to be used in early stages and less successful in late stages? Have you tested that?
Right. Well, you're very right with that. When you have a dramatic keratitis like the one Dr. Roth showed in the whole cornea, I don't think that would be a good case. I think early cases are probably the most recommended. And the good thing is that you can start, when you see first the patient, you can start with this. And of course, give the standard treatment as well. But you can, at the very first minute, try the photodynamic therapy, and that will give you a little bit of peace of mind that you're reducing the bacterial or the fungi load or the parasitic load. But you're right. I don't think that in bacteria that have penetrated really deep into the stroma, it might be as efficient. Yeah.
When we had a little lecture from Dr. Hammond yesterday morning, and he reviewed the clinical applications of the photodynamic therapy, and many use this high energies, and then they only using that for 3 minutes, and they claim, also Dr. Roth said this might be the key for success, but as far as I understand you, there is no time in these 3 minutes that the oxygen comes to the place where it's needed. So right. I think you are a fan of the classical long time treatment.
I love Professor Seiler's, patience with 30 minutes, because there's a law, a physics law, that actually we recently published a paper that limits the effect over time and in the depth of penetration. So if you try to do, it's not linear. You do more power or more concentration, and you're going to do better effect. It doesn't work like that. So it has to be constant exposure for a longer period of time to really get the killing effect. It's not a linear function, so that is very important to understand. And I don't think, we have done it in the lab, and it doesn't work. We published a paper recently about that.
What are you using clinically at this time before you get the FDA approval for your new concept?
Well, right now, this is not FDA approved, so it's an off-label use of cross-linking. We're not using or personally, I am not using cross-linking for infections right now. We're still doing the traditional method, but it's an off-label use. As FDA approved it, we can still, use it. But like I said, you saw the results. Sometimes the cross-linking doesn't work at all.
Yet?
I know that you're very lenient with the timetable, so thank you very much for that, Berthold.
You are as well.
Yeah, I know that. We're on the same level there. Two more comments. One question is, if oxygen is the key mechanism here, oxygen radicals, oxygen is depleted rapidly during the process of cross-linking. 30 minutes, you only need the first 3 minutes and then the oxygen is gone. It may be different with hydroxide
peroxide
supply.
But remember that you're putting the drops periodically during the hold, so you're giving more every time that you instill the drops. So that's the difference.
And you're doing the same during your process. You add the oxygen.
Yeah, we're doing the same as cross-linking, like every 5 minutes.
The question is, do you see a corneal refractive change?
Oh, no. We haven't gone into that. Well, this is again, I say this because you're my people, so and I can't disclose this, but potentially there might be a big effect in cross-linking. It might potentiate it probably, if it does with the antibacterial, probably 10 times. And it's been described. So.
Okay, we had 7 minutes discussion. Thank you very much.
Thank you so much. Appreciate it, guys.